Thanks to the organizers for putting together a great symposium so far. I'd like to touch on some of the themes that I've seen in earlier talks, thinking about long-term, short-term and long-term changes in the natural environment. And I want to do that by acknowledging the land that we sit on here in Montreal and in my university at Queen's in Kingston. And of course, this area was occupied by First Nations peoples long before the voyageurs canoed up the Great Lakes and settled the West, long before Christopher Columbus sailed across North America. And I think reflecting on some of this history has helped me as a scientist to grow and think about things that are actually important to science too, like the value of experiential knowledge, and thinking about things like baselines and for conservation biology. And also want to acknowledge the large and wonderful lab that we have at Queen's University, and the diverse perspectives that students have brought to the lab. And that's really helped to get me grow again as a researcher. And of course, our funding sources I want to acknowledge as well as an important driver of some of the research that we're doing. And so we work primarily on invasive species, but I'm more and more seeing these as just one sort of point on a perspective or a continuum of outcomes from extinctions to invasions. And in particular, I'd like to reflect on some of the genetic factors involved in this range of outcomes. And we like to do that by focusing on this parameter Darwinian fitness, which is a very simple thing to measure in natural populations, but it's related to a number of important parameters in population dynamics, in vital rates, and of course in evolutionary dynamics, and in particular the price equation, which I think is one of the most wonderful pieces of math and theory in all of biology. And so the question I want to address is related to this idea of the genetic basis or the genetic factors that contribute to a species distribution, not just in terms of range limits, but in terms of the variation in abundance and population growth rates that we see across the range. And because I know a lot of you are just coming back from lunch, I will give you the punchline, and then you can zone out for the rest of the talk. So the basic idea is that, at least from my perspective as someone who works with empirical data and interacts with a lot of conservation people, I think the dominant view of population genetics, range limits, and the contribution of genetics to species distributions is based around this classic view in which the landscape has a variety of optima. Populations are trying to reach those optima, but they're limited either by swamping effects of gene flow or the accumulation of deleterious alleles through drift. And I want to sort of reinvigorate this old idea of genostasis, which I'll argue, at least, that this sort of reconciles, sort of a way of reconciling the ecological idea of the species niche with contemporary evolution, that it can help to explain how we get maintenance of range limits at all levels of gene flow, and I think how it aligns better with some of the empirical observations we have in natural systems. And the real bottom line is that I think we need not just a theory of adaptation, but a theory of constraint, a better theory of constraint or constraints for understanding the genetic effects on natural populations. And so if we think about the sort of classic model here, we have this core population. It's adapted to its local optimum theta, and then the edge populations want to adapt to a different optimum, but they're constrained by the swamping effects of gene flow. That's sort of the outbreeding depression model, too much gene flow. Or the converse, which is those populations are small and edge populations are small, accumulate deleterious mutations, and as a result, they can't reach that optimum. So it's sort of like a Goldilocks, too much or too little, but what about the intermediate levels of gene flow? And so just taking a step back from these gene flow models and just thinking about sort of the fundamental model underlying local adaptation, we can think of it either as a range of phenotypes where we have two different optima, or we can think about it as an environmental gradient and the locally adapted genotypes at each point, at 2 points along that gradient. And a point I'll come back to is that some of these parameters, like the variance or the strength of stabilizing selection and the maximum growth rate that genotype can achieve, are generally considered fixed across that gradient. And so just, I'm going to go through this very quickly. You might want to have your cell phones ready if you want to take a photo of the reference slide that I'm going to show, suggesting how these, or why these predictions are not well supported in the data. But basically, some of the key predictions of this model, we expect stabilizing selection to be strong within populations, but divergent or directional between environments. If we do sort of reciprocal transplant where we grow genotypes reciprocally in each environment, we expect that the home genotype should do better when it's grown at home than when it's grown away, and we expect the local genotypes to do better than the foreign genotypes. If we do this for SNPs or alleles, we expect antagonistic pleiotropy. It's the same pattern, it's just we're talking about whether we're talking about alleles or genotypes. And so this is the slide with all the references, mostly meta-analyses, reviews. You might be familiar with these anyway. But basically, yes, local genotypes often do better than than foreign genotypes, but often things do just as well or better when they move to different sites. Stabilizing selection within populations tends to be weak, and divergent selection among populations is often selection is just directional. For example, flowering time is the reference here. Almost everywhere you look at it, there's directional selection for flowering time. And same thing with antagonistic pleiotropy at the genetic level. Even though it should be common, we mostly see conditional neutrality, where there's a fitness differential only in one environment, but not the other. And so that brings me to the idea of genostasis. So Janis Antoniewicz, decades ago, pointed out or argued that genetic variability or genetic variance, what we would call today, and the swamping effects of gene flow are inadequate to explain limits of natural selection. Toby Bradshaw in 1991 coined the term genostasis as the term for this phenomenon, which is not just about genetic variance, but the appropriate type of genetic variability. And several recent studies have sort of taken a multivariate approach to this to suggest that even though individual traits may be highly heritable, the extra dimensions, when you start thinking about trait correlations and the many different traits involved in an adaptation, there can be constraints at those higher levels. So very quickly, these work that we've done on purple loosestrife is more consistent with the genostasis model. And very briefly, the idea is that if you, when we grow populations in a northern environment at the top left, or in the southern environment at the bottom left, we find that selection is pretty much the same, which is that selection favors early plants that flower early but are larger, but the development of the plant, the biology of the plant restricts development along that principal component axis. And so as we go into northern environments with shorter growing seasons, the environment gets more stressful, and that ratchets down the available genetic variance, the optimum phenotype, and the maximum fitness that the population can achieve. And if you think about this more generally, there are many biotic and abiotic stress factors that plants and animals are dealing with in nature. And so how do we modify this sort of classic view of local adaptation to include the genostasis idea? We just relax assumptions about constant variance or stabilizing selection and a constant maximum growth rate. And so if you just focus on the right column there, you can see the main difference here. We're talking about two genotypes. Under the genostasis model, the optimum environment is the same, which is the low stress environment. And they differ in this trade-off, either having higher fitness with G1, having higher fitness in the benign environment, or maintaining fitness over a broader range of environments, as you see in the G2 blue genotype. And so just very quickly, to try to wrap up in the time, which saying that has actually thrown me off. So anyway, the dashed line here, just think of that as the species niche, but within the species we have different genotypes shown by the colored lines. Of course, the overall variance in fitness declines along the gradient, but if we look at the variance in relative fitness in that bottom right, there's a minimum at intermediate stress levels. And the key point here is that each of those genotypes have the same fitness across the gradient. If you look at all the locations across the gradient, even though at any individual point along the gradient, there's a strong fitness differential. I'll skip over this, but it's just about how, just if you look at the points and imagine this as a reciprocal transplant experiment, some genotypes might show local adaptations, others might not. We can extend this to to multivariate stress gradients. So here we're thinking about an abiotic versus a biotic stress gradient. We consider common gardens in each of those 4 locations. And you can see that any pair comparison of a reciprocal transplant experiment, there's only one case where we would actually detect that antagonistic pleiotropy or that classic crossing of reaction norm. And some of this is published, some of this we're still working on, but I just want to draw your attention to this paper, which gets into some of that multidimensional stress gradient stuff in more detail. And I'll just leave you with this final slide saying, again, reiterating that I think we need a better and stronger theory of constraint in order to understand the future response of plants and animals to climate change. Thanks. Sure.
Could you also get the same pattern? So we have a stress constraint model, but with like a cost to it between phenotypes? Wouldn't that produce like a similar pattern?
Yes. So the question is about if you get a similar pattern with a cost of extreme. Yeah. So in this case, the cost is in terms of the trade-off between fitness at low stress versus high stress. But you could get, like if there's no cost in the benign environment, is that what I'm understanding? Yeah. I didn't explain that well, but yeah. All right, sorry I went through that so quickly, but thanks.
Our next speaker is Marion Thaureau. Exploring phenotypic evolution as a consequence of changes in the adaptive landscape across lineages and timescales.
So hi everyone, thanks for being here today. I'm very excited to present this project, part of my PhD, which aims to better understand phenotypic adaptation to environmental changes across timescale. Can you hear me? Is it? Yeah, perfect. Okay. So in evolution, usually we can look at the pattern at different timescales, and in macroevolution, so the big timescale, we have events such as speciation or extinction, while when we are looking at the patterns at smaller timescale, we have the processes such as selection, mutation, or also genetic drift, and these processes should explain what we observe on the bigger timescale. However, a lot of studies have demonstrated that on the long timescale, we have a lot of stasis with slow changes, while on the very small timescale, we have drastic changes and a lot of movement, adaptation, and fast evolution. So we want to better understand how these patterns occur at the different timescales. And it has been suggested by J.J. Simpson in 1940s that adaptive landscape might be this conceptual bridge between the different timescales. So today we want to better characterize this adaptive landscape at the different timescales. And for this, we decided to take 3 different data sets across timescales, so representing different timescales. So first we have phenotypic time series. For the very small time scale, we have the evolution of the body mass in a population of salmon across a few decades. And then for a bigger time scale, we have the length variation in a species of coccoliths over a few 100,000 of years. And for the biggest time scale, we have the evolution of the length in a lineage of foraminiferas over millions of years. So because we don't have so much time today, I will just focus on the smaller and the biggest time scale and talk less about the coccoliths. But for each of these phenotypic time series, we also have environmental time series. So we have the evolution of the river water flow for the salmon time series, and we have the oxygen and carbon isotope variations for our coccolith and the foraminiferas. And all these time series have been already published, and it has been demonstrated that we observe correlation between the phenotypes and the environment. But today we are mainly interested in seeing if there is a causality, so if truly the environment is influencing our phenotypes. So how are we doing that? I'm sure you all have this question in mind. So we are going to fit a lot of different evolutionary models, and they are mainly from two types to simplify. So first we have the unbiased random walk, so a random evolution of our trait around the mean. And the other type of model that we have is called Ornstein-Uhlenbeck model. We have the evolution of our trait toward an optimum. And is in this case, so we have the phenotype in blue, and the optimum is here fixed in orange. In our study, we are using two novelties. So first here, the optimum is fixed, and this optimum is the value of the trait maximizing the fitness. But we know that this can depend on the environment, on the abiotic and biotic factors. So we allow for movement in our optimum. So therefore, the phenotype has to constantly readapt to a changing optimum. And then the second thing that we are doing with this study is that we are using this time series in a multivariate framework, meaning that we are allowing for more than one time series to be analyzed in our models. So therefore, we can study the relationship. So first, yes, the correlation between two time series or more than two, but also the causality. So if one of the time series is truly influencing the movement in the optimum of another time series. So the question is, how do we parameterize this model? And if you are not very familiar with the OU model, we're going to see a few slides on this. So here is the equation describing the OU, so the non-random processes. And there is two terms to this equation. So in darker blue here, I'm not sure if you can see the pointer, but in darker blue, we have the random noise, so the stochastic part describing our time series. And in lighter blue, here we have what we call the deterministic part, so which show us the non-random evolution. So what is interesting for us is that with this part, we can see if there is a causality and if our time series is evolving toward an optimum. So we are going to focus on this deterministic part. And the way we are modelizing it in our scripts is by using this kind of matrix, so which is called a matrix of deterministic variations. And in the diagonal, we have like the deterministic variation of our trait and the deterministic variation of our environment, while in off-diagonal element, we have the influence of the environment on our trait and the influence on our trait on the environment, which is less common but can happen in some environments. So if we have 0, that means that our value doesn't exist in this specific model. If we have a 1, that means that this parameter exists in our model. So here, for example, this matrix describes an environment evolving randomly, because we have a 0 here, while the trait is evolving toward an optimum. And here, because in the off-diagonal element we have a 0, that means that we have no influence of our environment on our trait. But we can have different type of this matrix. So here, for example, we still have our environment evolving randomly, and our trait is still evolving toward an optimum. But here it's a bit different because we have this parameter, which means that our environment is influencing our phenotype. So I promise it's going to be useful for the result part. So now the question we want to know is how we are using this model to better understand how species, lineages, and population are adapting. And first, we fitted these different models at the micro-evolutionary scale, so in our salmon dataset. So to talk a bit about this time series, this is a population of salmon living in a river in Norway. And over a few decades, we have a drastic decrease of the body mass going from 12 kilograms on average in the 1920s to nowadays 4 kilograms. So this is a very big decrease. And meanwhile, a lot of dams, like 3 dams at different periods, have been constructed on this river, decreasing also drastically the river water flow. So it has been shown in this paper from Jensen et al. That we have a correlation between the body mass of the salmon and the river water flow. So with our model, we want to see if there is a causality, so if we can really see the influence of the environment. So here you have our time series. In blue, we have the phenotypic time series, the body mass of the salmon, and in green, the river water flow. And we fitted a bunch of different evolutionary models, and trust me, this is only some of them. And we compared the AIC to find what was the best model. And here, this is our best model, which corresponds to this deterministic matrix. So if you remember what I said during the explanation of this kind of matrix, here we have our environment evolving randomly, while our trait is evolving toward an optimum. And we have also this number here showing that we have a big influence actually of the environment, so of the water flow on our salmon body mass. We also have the half-life here of 1.6 years, which is the half of the time to evolve toward the optimum of the salmon body mass. And this half-life is very short, indicating that the influence is also very strong. So now we want to fit the same models with our macro timescales time series. So we have here the evolution of the length in the lineage Globorotalia across 3 million years. And here are our time series. So at the bottom in blue, we have the foraminiferal length. And then in green, we have the evolution of the oxygen and the carbon isotopes. And we want to see if we observe the same kind of pattern, if we can also recover the causality of 1 or 2 environments on our phenotypes. So here are our best model, first for the length of the foraminifera and the oxygen isotope variation, and also for the length and the carbon isotope. And in both cases, we recover the same type of deterministic matrix that you might recognize from the salmon. It's exactly the same type of result. So first, we have our environment evolving randomly, while our trait is evolving toward an optimum. And you can also see that in both cases, the oxygen and the carbon are having a strong influence on the length of our foraminiferas. Yeah, also the half-life indicates to us that the oxygen has a bigger impact on the trait, but still both of them are playing a role. You can also notice that we have a negative causality for the carbon, so it's evolving toward the different opposite direction. So for the small story behind this, it has been suggested that this evolution of the oxygen isotope, so this increase could actually reflect the migration of the foraminifera lineage toward deeper layer in the ocean. So therefore, the foraminifera will have to adapt. And by increasing its size, it's actually reducing its ratio surface area, which is also decreasing its buoyancy to make it able to adapt to a deeper layer in the oceans. So what I want to emphasize with this study is that the adaptive landscape might be more dynamic than suggested, especially for the macroevolution, because it's often described as stasis. And the models using moving optimum might be very important to better understand the processes of microevolution and how they are leading to these macroevolution patterns, which will help us, hopefully, to better predict evolution. So yeah, and to finish, I would like to thank my supervisor, Kjetil Voyeux, that unfortunately cannot be here, and all my lab at the Natural History Museum of Oslo. Thank you so much for your attention.
Our next speaker is Kevin on assisted evolution in corals.
Thanks everyone for coming. I'd just like to start by acknowledging the land that we gather on. I'd just like to pay my respects to all elders past, present, and emerging. So I know you all probably came for pretty photos of coral reefs, and there's a half truth to that in that I'm a terrible photographer, and sadly scenes like this are kind of disappearing, at least the diversity of corals, as you probably already know. So just based off of calcification recreated temperatures, we can see that the temperature anomalies today have risen quite a lot. And we're seeing a lot more mass bleaching events of coral around the world, including the Great Barrier Reef, where I work, where we've seen 5 mass bleaching events, including this year, in the past 9 years. 5 out of 9 years we've seen mass bleaching events. So it's getting quite a lot of bleaching events. But luckily, there's not all bad news in that we've already seen, even in up to 2017, we've seen responses to this warming. We've seen increasing thresholds of bleaching temperatures for many different coral. And obviously, this response is both through acclimation and adaptation, but there are many different studies highlighting the polygenic and heritable nature of bleaching resistance in corals, including many at this conference. So there is potentially some hope. So the question really becomes, will coral adaptation be fast enough to keep pace with future expected climate change? And the answer is, based on models, possibly not. This is a model that recently includes a very simple way of doing evolution using the Breeder's equation, where we see that with increasingly pessimistic climate scenarios, we see increasingly pessimistic population outcomes as well. So in the next 100 years, we're not sure. It's kind of it's anyone's guess whether or not they're going to survive, but it's not looking great in terms of the modeling perspectives, and many models have the same result. So if not, can we at least try to stave off or increase the genetic variance in the population, or assist population adaptation in the interim while we get climate change under control, hopefully? So that's where this kind of project comes in. I'm going to be talking to you today about 3 different assisted evolution interventions. They are in no way complete. There's many more interventions out there. So I just want you to know that, and there's many different ways of doing them. The first one we can consider, which has already been done today in many places, but not specifically to resistant corals, is coral fragmentation, where if we have a kind of a heat stress event where many different corals die, we can use the ones that are resistant and surviving bleaching events, fragment them, replant them, and grow them up into adult colonies. And this is thought that maybe we can to confer their resistance. So clonal fragmentation, obviously using clones. And this is obviously done in many parts of the world. Now it's being increasingly focused on selecting the coral that are resistant to bleaching. We've also got assisted evolutions in terms of selective breeding that are becoming a lot more considered, and actually almost in terms of implementation phase being more scalable in terms of selective breeding, where we take the bleaching resistant colonies, we merge their gametes, and we can, since they produce hundreds of thousands of gametes, we can take these pools and create many, many recruits. And we can grow them to adult size, and this can avoid some of the size-dependent survival pressures as well. So here we are collecting natural gametes in the field, as well as growing them in, this is the Australian Institute of Marine Science here. And finally, a very futuristic one that obviously is not practical or morally acceptable today is gene editing, but we have to consider that in the future, potentially, maybe, we could be able to implant a single allele of large effect, is how we've thought of it, into newborn corals, and then they could grow up to be thermally tolerant. So this would just be a random selection of corals in this case. But potentially, we could have this technology into the future. And we've already seen some studies of CRISPR-Cas9. It just has not been applied in terms of a restoration perspective. So I also considered this hypothetical future assisted evolution intervention. So the question becomes, which intervention is best for adaptation? Here's where I want audience participation. If you could just raise your hand if you think that clonal fragmentation would be viable and work. And I've already done this, I should say, I've done this poll to a coral conference, so it's going to be interesting to see it at an evolutionary conference to see the differences in results. Who thinks clonal fragmentation? Wow, so many people. No, I'm just kidding. And who thinks selective breeding? Lots of selective breeding. And who thinks gene editing, if it were feasible and morally acceptable? Okay, so I definitely think the selective breeding kind of wins out in terms of the crowd opinion. I'm also going to sneak in here a little side thing that I'm going to consider many different allele effect sizes, so it kind of gives gene editing a little bit of an advantage. All right, so to evaluate these interventions, we need to think of a hypothetical place that I call Fantasy Island, where we have, a beautiful coral reef like this, where we have infinite money to run these experiments, infinite time to run these experiments, and most of all, infinite computational power. And so this is where we come into modeling. So here I'm going to simulate 100 across, we're going to assume that there's 100 QTLs, adaptive loci, that can be selected for in a non-Wright Fisher population model of corals. And it's a fairly large population size, I should mention, hence this choice of 100, because it's almost 1 million individuals, so it's quite a cumbersome model already. And we're going to be looking at determining which interventions increase population size the most relative to doing nothing in terms of future warming. And to do this, I've used Ben's wonderful software, SLIM, to do forward genetic simulation and try to understand what will occur with these 100 loci. All right, so where is this fantasy island? I've selected Moore Reef in the Great Barrier Reef, mainly because it's very well studied as the kind of study site. And here I'm using downscaled climate projections using a middle-of-the-road or SSP 2.45 climate scenario. And from this, I've also got baseline or like historical temperatures that I've used to inform the phenotype, just assuming that corals are adapted to their baseline phenotypes from that. So how the model works is we've got a population, and every individual within this population has 100 QTLs on both chromosomes. We sum these 100 QTLs together to get a breeding value or genotype of these individuals. This creates the additive genetic variation, or the distribution of additive genetic effects, and thus variation. We can also use that the baseline V temperature to get our environmental effects just using the Breeder's equation, assuming that that's how they're adapted. And then we randomly draw an environment effect for every individual, which gives us the individual phenotype, which then goes back into the overall population phenotype distribution. And so this is kind of the genetic side of the model, using the quantitative genetic animal model as a framework for this. And corals obviously track their environmental temperature. So to do this, for instance, consider a coral that's adapted to 25 degrees Celsius optimum, and they're experiencing a current temperature of 28 degrees Celsius. So they have a mismatch of -3. Given this equation, it could be a probability of about 50%, which is basically we're flipping a coin to see whether or not they survive to the next year. So it is going to be an absolute survival whether or not they do survive. And finally, I'm not going to talk about too much, but we've got varying demographic rates in terms of survival, growth, and reproduction, as well in terms of coral size, which is really strong demographic effect on coral, basically. All right, so we run the simulation. What do we get? So we get a lot of squiggly line graphs like this, and so I'll just go through just kind of what's going on here. I'll just try to do it by myself. So we've done 30 different genetic seeds. And we've got here the no intervention versus selective breeding. And that gray bar is where we've applied the intervention across 5 years. And we've replaced 10% of the natural recruitment. Obviously, this plot is really hard to read, so I'm just going to convert it into percent difference for you, so it's a lot easier to see. So if we just look at after intervention, what we see with selective breeding is that there's a net positive effect. So the top of the graph being a positive effect on population size overall due to selective breeding. So that's just one of the interventions. If we add the other two, we can see that the best genetic editing increases it even more. If we have a genetic editing where we have an allele of about 0.5 degrees Celsius, it increases it even more. But clonal fragmentation is a lot less, and I'd like to highlight that there's because we're replacing 10% of all the recruits, the amount of colonies that you have to fragment and break apart ends up just plummeting the population size because you have this insane influx of recruits. So obviously that's possibly not realistic at all, but generally we see that it's not going to be the same at all compared to selective breeding, and we're going to have to kill a lot of healthy resistant adults to do so. And there's also an effect of actual, that I've seen of actual, just the genetic variance decreasing as a result of clonal fragmentation as well, apart from that mortality effect. If we just look further into the effects of genetic editing in terms of just varying genetic editing, we see that there's kind of a happy medium that does the best. And then if we go too far, it actually selects out that allele, where basically if you have a really strong allele and it's too early essentially, it's not adaptive yet, it's just selected out of the population. So we can go too far too fast, we also see with genetic editing. Obviously, if you're too adapted, you're also maladaptive in this model. And then just looking at a kind of a complicated graph of the mean genotype. So now we've got kind of the mean genotype of the population versus the change in population size. I'll try and reset at the right time. Well, I'll just, and then we've got this kind of what I call evolutionary rescue. It's not true evolutionary rescue, but this is kind of changing both the genotype and increasing the population size at the same time. We can look at the kind of 4 select interventions here, and we see that the gene editing kind of goes towards what I would call more evolutionary rescue, where it's both changing in terms of the mean genotype and the population size, but then selective breeding, it kind of jumps really quickly up, but then it actually travels backwards. It's actually losing the mean genotype over time. So I'm not sure exactly what's happening here. So I'm interested in any thoughts on kind of theory to explain this process. So if you have any ideas, I'd love to know. So yeah, selective breeding improves population size early on, but then it's not maintained in the long term. So it is still it's still beneficial. It's still helping the population a lot. It's just the genotype is regressing backwards over time. All right. So we've seen these different interventions. It seems like clonal fragmentation, as you guessed in the audience, it's not so viable compared to the other two. And obviously, gene editing doesn't exist yet, and it's not really morally acceptable. Especially traditional owner groups do not like gene editing, so it's likely not going to be available anytime soon, or used anytime soon. And we can go too far. We really need to know exactly how far we're adapting them if it did exist. And this is likely to also occur for selective breeding. If we could enhance it even more, it's likely that we could go too far. So there really is kind of a Goldilocks type effect here. We have to be kind of in the middle of this. Final thoughts, don't trust the exact population numbers. They are untrustworthy. This is my model versus reality. It's I'm missing a lot. I'm missing many other, obviously, eco-evolutionary processes. We don't exactly know the number of genetic parameters, and it's an overly kind of optimistic now climate scenario, so we're not sure. It could actually be a lot worse than this, potentially, so there might be even more extinction. But we can kind of qualitatively say which of these interventions seems to be working better, and it really seems like clonal fragmentation is not going to be viable, at least the way we've implemented it. Finally, selective breeding can be a lot more effective if we consider assisted gene flow. So it could actually go a lot more like a gene editing, where we actually could overdo it if we have too much assisted gene flow, which is what I plan to tackle next with this model, but haven't done so yet. Yeah, thanks for listening, and if you are interested in another talk, go check out Eva's talk as well. She's coming up tomorrow. Thank you.
Our next speaker is Thomas on multivariate analyses.
Hi, I'm Thomas. I'm a PhD student in the Stepanek lab at the University of Toronto, and I'll be talking about the conditions in which a P-matrix can be used instead of a G-matrix for making evolutionary predictions. So predicting evolution in quantitative traits has a long history in plant and animal breeding. We use it to breed economically valuable traits. We can also use this to predict responses to selection in experimental evolution in the lab, or we can also use it to predict whether endangered species, such as a spotted turtle, will be able to respond appropriately to selection in its environment. Predicting evolution in quantitative traits can be done using the multivariate breeders equation. So here we have a change in a trait mean. So this is a multivariate trait mean, and that is due to a selection gradient beta times the genetic variance covariance matrix. The genetic covariance matrix gives you the distribution of additive genetic effects in a population. So here I have an example of two traits, trait 1 and 2, each having a genetic variance of 1. Those are the diagonal elements of the matrix. And then the off-diagonal of the matrix is 0.5, meaning that the genetic covariance between these two traits is 0.5. So we get a distribution with a moderate correlation between the additive genetic effects of these two traits. So this is central to predicting multivariate evolution. It gives you the directions in multivariate trait space where a responsive selection is possible or may not be possible. But estimating G requires us to decompose phenotypic variance into its genetic and environmental components. Typically, we do this by fitting a linear mix model, and this requires genetic information. So we can either use a pedigree, which you might use if you have some kind of breeding design, such as in agriculture in the lab, or if you have some kind of wild population, you might use genomic relatedness matrices. This comes with some difficulties in many situations where you might want to predict or reconstruct evolution, especially if you have no genetic information. So studies of fossilized populations, you're not going to get any genetic information out of that, so you don't have a G matrix. You might have small wild populations, which are very difficult to estimate G matrices for, or organisms with long generation times. G matrices also come with large sampling error, and this will introduce error into your evolutionary predictions. A common solution for these problems is to use the phenotypic covariance matrix P as a replacement for the genetic covariance matrix. In 1988, Chevre suggested that phenotypic information is easier to acquire than genetic information, and it comes with a smaller error. He also suggested that P matrices will often resemble G matrices because of developmental processes, and therefore that P matrices may provide more accurate evolutionary predictions than G in some situations. This is done by taking that multivariate breeder's equation on the left side and then replacing the G matrix with a P matrix to get your approximated evolutionary response. So again, this is a commonly used solution in situations where G matrices are just difficult or impossible to estimate. But the P matrix is the sum of both genetic and environmental covariance matrices. So you have to consider the distribution of environmental effects as well. So you can see from this equation already that your evolutionary predictions may be overestimated in magnitude from the P matrix, but it is less clear about how that might impact directions of evolutionary responses. So previous tests of Chevre's conjecture, such as this Roth 95 and Dr. Min 2011 papers, will often take pairwise comparisons of phenotypic and genetic correlations and see how similar they are to each other. So you have on both these plots phenotypic correlations on the x-axis, and on the y-axis you have their corresponding genetic correlations. And this is done for a range of different animals and sexes in different environments, and we often see a close to one-to-one relationship between these two types of correlations. But these previous tests do come with some limitations. Empirical data sets are often used, and because of the large sampling error in these estimated G matrices, that's going to have some input of error into these comparisons. We're also comparing pairwise correlations in these previous examples instead of the covariances, which are what is actually determining evolutionary responses. Oh yeah, we're also missing some multivariate structure of the matrices when you take these pairwise correlations. And an example of this, we have basically distributions of phenotypic and genetic variance here. So these pairwise covariance comparisons are not sensitive to the multivariate properties of these matrices. So on the left side, we have distribution of eigenvalues for a P matrix. Commonly, they will have some variance for each principal component of the covariance matrix, while a corresponding G matrix will have what we call these null subspaces. So these null subspaces are basically directions in phenotype space where it's impossible to have an evolutionary response because there's a genetic constraint. So there's many conditions in which using a P matrix will give you an evolutionary prediction of response in some direction, which isn't actually possible because of genetic constraints. So to answer when P can substitute G, we take a simulation-based approach. We simulate breeding designs with various parameter combinations to produce data sets with estimated G matrices, true parameter G matrices, and then the corresponding P matrices. And then we use multivariate comparison techniques to see how similar these matrices are to each other. Simulation parameters start off with different multicollinearity. This is basically how we set the distribution of genetic variance in phenotype space. So if multicollinearity is low, we have genetic variance kind of in all directions. But if we have high multicollinearity, then variance is kind of loaded onto the maybe first principal component, while the other directions in space have very little variance. We also vary heritability between 0.1 and 0.5, and then the number of families in the pedigree ranging from 50 to 500. We also vary the distribution of environmental effects relative to genetic effects. So we can have proportional environmental effects to genetic effects. They might be independent to each other or in opposite directions to each other. In red, I've highlighted the situation in which Chevre kind of envisioned this optimal condition of P approximating G. So to start off with our analysis of our simulated data set, we start where many other papers have left off in these kind of pairwise correlation comparisons. So on our x-axis, we have true genetic correlations, and then our y-axis, we have either estimated genetic correlations or phenotypic correlations. So in the red, we have our estimated G compared to our true G, and then on our right, we have our P matrix correlations compared to our G matrix correlations. So we see that when environmental effects are proportional to genetic effects, we get a very close one-to-one relationship between genetic and phenotypic correlations with small error. But when we change the distribution of the environmental effects, we can get underestimation of genetic correlations if you were to use phenotypic correlations as an approximation. When environmental effects are opposite to genetic effects, there's very little relationship between these correlations. But to move on to our actual multivariate comparisons, we start off with what's called common subspace similarities. And this is a measure of how similar the orientations of variance are between two covariance matrices, measured on a scale from 0 to 1. So on our left side, we have an example where the similarity score is 1. So we have two matrices that are aligned in the same directions. But if they're perpendicular to each other, like in the right side, then we're going to get a similarity score of 0. So here on our x-axis, we have effective sample size, which we define as, or Shevard defined as number of families times heritability. And our y-axis, we have common subspace similarity. So the closer these points are to 1, that means that we're getting a better estimation of orientation of genetic variance. So when environmental effects are proportional to genetic effects, we see that the orientation of genetic variance is estimated well by the P matrices and the G matrices. So we have in red the common subspace similarity between the estimated G and the true G, and in blue we have the P and G comparisons. But when we move to independent environmental effects to genetic effects, or if they're opposite to each other, we see how low heritability creates these situations where P matrices are not estimating these orientations of genetic variance very well. Our next multivariate comparison technique is called random skewer correlations, and this is to assess how similar our evolutionary predictions are between our G and P matrices. So recalling that multivariate breeder's equation, we sub out, we take both the estimate from the G matrix and the P matrix and then take the correlation between them in response to a selection gradient. We simulate 1,000 random selection gradients and then take that average. So our first set of comparisons, we have our high multicollinearity case. So again, we are basically having some directions in space where there's lots of genetic variance, and then in the rest of directions, there's not much variance. And we can see when environmental effects are proportional to genetic effects, we're getting very accurate estimations of evolutionary response using that P matrix. Especially even when effective sample sizes are smaller, we're getting more accurate estimates with P than we would with G. But again, switching to an environmental distribution that's independent to genetic effects or opposite, we see that the genetic covariance matrix provides more accurate evolutionary predictions. And that's especially true when heritability is low. So those little circles are low heritability cases. And we see that the blue points, the P matrices, are doing much worse at providing evolutionary predictions than the G matrix. We then move to low multicollinearity, so we have kind of an even distribution of genetic variance across phenotype space. And we see that consistently the P matrix provides more accurate evolutionary predictions than G, especially for low effective sample sizes. So this is a condition where, in general, a P matrix is going to provide a more accurate evolutionary prediction than G. So to answer the question, can we predict evolution with genetics? Well, predictions with P are most accurate when traits are genetically uncorrelated to each other, so low multicollinearity often. Otherwise, when environmental correlations are proportional to genetic correlations, those are conditions where a P matrix will perform the best at providing these multivariate evolutionary predictions. Large sample sizes are still going to be preferable for P, so despite their lower sampling error, you tend to get, sorry, I realized that's something that's probably about 100 families is my best guess, or individuals. Otherwise, the G matrix is generally preferable. So this all goes down to a bias variance trade-off. G matrices are going to be higher variance estimates, but there's going to be less variance, or less bias, while a P matrix will be biased by environmental effects, will have lower variance in its estimates. So our take-home message is that before using P as a substitute for G, consider the multicollinearity of your trait set. So higher levels of genetic correlation or integration will confound evolutionary predictions using P. In those cases, you should also consider the environmental correlations. So if they're proportional to genetic correlations, sometimes in these cases, P will provide more accurate evolutionary predictions than G. So I'd like to thank my supervisor, Jacqueline Stepanek, as well as the rest of the Stepanek lab, so Dr. Mathieu Vidilier, and the rest of the lab for help and support for this project. You can feel free to contact me at my email here, and I would also like to thank my funding sources, and I'll be happy to take any questions. Thank you.
Our next speaker is Andres Cortés on genomic prediction, and this is a video.
The motivation behind is to infer local adaptation in wild beans by looking at the environments where those beans come from. And this idea has been built since Humboldt's writings, in which he reflects that geography environments exerts its influence on plants. Therefore, nowadays we should be able to go back and infer how nature influenced the adaptation in those regions for the local ecotypes we find there. Under this idea is that we approach the current dataset, which involves wild beans across the America, the natural distribution range for the species. Wild beans are very diverse in terms of size and color in their seed coat, but they are also very diverse in terms of the environments they occupy. So when we look at the wild beans, which are blue dots in this map, and we try to look them across the Americas by looking at the precipitation in those regions, the annual precipitation, we might start seeing that Mexican wild beans tend to be in places where there is more permanently dry environments compared to beans that come from South America, such as Colombia, Ecuador, or not Peru. This is just by looking at the precipitation, but of course drought stress is perceived as a balance in which water not only comes into a system by precipitation, but it's also loosed from the system by evaporation and transpiration. So that's why we do not only look at the annual precipitation patterns, which are summarized here, but also try to look at a couple of indices that combine those two properties, how much water the system receives, how much water the system loses through evaporation and transpiration. And by looking at this drought index, we might find again that beans from Mexico tend to be apparently more drought adapted than beans from Colombia and South America. So this data set, which is, as you can see, built not on raw bioclimatic variables, but instead on ecophysiological models, is what enables us to build a predictive model on the local adaptations in those sites. And again, I'm insisting we are working on wild beans because for those ones, it's possible to assume that competitive testing has been testing those plants more than persistence. Persistence is the case of the cultivated beans that are probably in these regions because farmers brought them very recently, as compared to wild beans that have been under competitive testing, in this case for drought adaptation during many generations. So based on that assumption is that we performed this first genome-environment association, looking first at drought tolerance, computed by the indices I just showed you. And interestingly, we see here marked in red the different regions and SNP markers that associate with the drought index inferred with environmental variables. And if we move across chromosome until the 11th chromosome, you might realize that this system is highly polygenic, meaning there are many regions with low to moderate effects influence the adaptation to drought tolerance in wild beans. This brings us different questions, such as how this system arose in first place. Was it perhaps a mutation that later on served as a platform for other mutations to build upon? And also, how can we predict adaptation when the system is so polygenic, such as this one? So we will explore these questions, but before I would like you to reflect on the nature of this variation and what's perhaps the driver behind this adaptation. And by that, we compare different summary statistics between associated and non-associated regions. Associated regions tend to be more in highly SNP density windows, also with slightly high nucleotide diversity, lower theta Watterson's statistic, and the combination of these two leads that associated regions are generally under higher Tajimas-D scores, which can be interpreted taking into account that the mapping exercise and this exercise use population structure as covariables to divergent selection towards drought tolerance. Also look at this. This can be interpreted as a background selection across the genome, any type of signature associated, for instance, with adaptation mainly derived from the gene pool structure that is observed in the species. In other words, this conclusion leads that drought tolerance might be beneficial under drought stress, but as expected, is not necessarily so good under well-irrigated environments. And this is the typical trade-off we would expect for a locally adapted trait. Genome-environment associations, as I said, are controlling for potential spurious factors such as demography and the background selection in the non-associated regions. But what happened when we look not just at drought tolerance, which concluding seems very polygenic and being under divergent selection, but instead we look, for instance, to heat tolerance in the same panel. In this case, the index was built more in a more simple way. It was simply in the cases in which temperature in the evening was higher above a physiological threshold for the species, and that is because if the threshold goes, the temperature in the evening goes above the threshold, the pollen loses viability. So we map again those regions across the 12 chromosomes, across, using different algorithms to inform the genome-environment associations here in different colors. And again, we find ourselves with a highly polygenic landscape, which does not necessarily correlate, as we have explicitly carried out, with the previous one. So it's not a case of a polygenic pleiotropic adaptation. It's completely different adaptive data sets that are being involved in this combined response. Here in red asterisk, we see genes that have been previously described as heat shock proteins and that correlate with these associated regions. So this also serves as an ad hoc validation for this analysis. So we have these two scenarios in which we have adaptation understood as drought tolerance, as heat tolerance across wild beings in the Americas. We try to understand a bit how it was built, and it's related with this divergent adaptation across environment, which implies a trade-off for adaptation and yield, which is what perhaps sometimes is more interesting for future applications. But still we have a remaining question, and is how can we predict adaptation in the system? And here is what we are currently doing, and that kind of pushes the field forward, and is trying to estimate genomic estimate adaptive values as a proxy to what before was known as regions of known adaptation, of prediction of known adaptation. Instead, genomic estimate adaptive values can be understood more related to what in plant breeding is called genomic estimate rating values, or in medicine is called polygenic risk score. So all those three are estimates that assume a polygenic landscape versus the risk of no adaptiveness, which assumes a more Mendelian determination of the adaptation. So here we move forward into a validation set, and we build interspecific hybrids between beans, common bean and tepary bean, being tepary bean known as our source of heat and drought tolerance. Those crosses were back-cross complementary and tested in 4 localities for 4 yield traits and across more than 15,000 SNP markers. Again, the combined landscape is highly polygenic across different localities, which are marked here in color and symbols. That meaning that not only we have a polygenic system, but it's a system that reflects a lot of genome-environment interaction across the regions. So building the prediction here is much more difficult if we rely on traditional methods, and that's why we move into these dynamic estimate adaptive values. Unsurprisingly, our predictive ability, that is how much correlation we found in our testing data set compared to the prediction, comparing the prediction and the observed real value, we are reaching for different stations, the multilinear stations, up to 75%. This is only for one of the 4 traits that was examined, each of these lines, but across different lines and colors, we have the different traits. Again, for another locality, nearly 75%. And if we zoom in into these regions, we reach the saturation very early on. With only 250 SNP markers, we are already reaching the saturation of the prediction. That meaning that we know adaptation in wild beans is very complex, involving a lot of regions, polygenic regions, and genotype environment interaction, but still we are able to successfully predict the adaptations in those regions by using genomic prediction, in this case a Bayes-C algorithm. Difference is that we are not estimating a breeding value, but instead an adaptive value. This led me to close with another citation. This book, Darwinian Agriculture, Natural Selection has already tested more combinations than humans ever will, and therefore, if we have a polygenic system such as this one, we might learn more by looking at the system and comparing it across regions and species rather than trying to replicate it. So as conclusions, genome-environment associations and genomic estimate adaptive value enable exploring and predicting the genetic basis of adaptation in wild beans. Both approaches utilizes mixed linear models as response variable. They are including not a phenotypic variable, but instead we suggest an environmental index as compared to a raw climatic variable. It is advisable, we insist, to use these physiological-based indices since they are more informative of the processes we are looking as part of the adaptive response. Combine that with summary statistics to interpret the cause behind those selection signatures and try to predict those responses using genomic prediction values seem to move forward the field from interpreting and staying on genome-environment associations and trying to leverage those materials as predictive scores. We have applied this, both genome-environment associations and genomic estimate adaptive value in common and temporary beans to describe the architecture to both draw and heat tolerance, but more importantly, to predict the potential of abiotic stress tolerance in the species. Thanks a lot.
So I'm going to introduce the next speaker, Moi, who will be talking about rapid evolution in Arabidopsis.
All right, great. Welcome back for the last session. Also, I hope that you stay for the last talk. We're going to have a panel discussion to discuss about rapid evolution under climate change. And today, I'm going to tell you about rapid evolution with our favorite model system, Arabidopsis. Our footprint on Earth through land use and climate change is accelerating the extinction of species. We've seen this paper cited several times. It's a summary of 150 studies trying to predict how many species might be under threat of extinction this century. But what we've highlighted several times is that none of these predictions, including evolution as a possible escape routes. And this is in part because we've always thought of evolution as a slow process that leads to the speciation. Whoop. Okay, that's a trick that I need to learn. Black and back. That leads to the speciation through the tree of life or something that we can study with fast-growing organisms like E. coli, yeast in laboratory conditions. Through the genomic revolution, some serendipitous experiments like the domestication of some species, some long-term did it again. Long-term monitoring of natural populations. We have realized that evolution can be rapid in higher eukaryotes thanks to the presence of standing genetic variation, pre-existing genetic variation. But we haven't done yet experiment, a principled long-term evolution experiment with one of these eukaryotes. And of course, we have to do it with Arabidopsis. Because apart from that is very beautiful, is broadly distributed geographically across many regions in the world. Presumably there is standing variation related to climate adaptation. It has unparalleled germplasms and genomic resources. If you want hundreds of genotypes, I'll send them to you with genomic data. So we can create a powerful long-term evolution experiment. So how would this look like? My ideal experiment would be we collect lots of seeds from across the geographic range to maximize that starting pre-existing genetic variation of a population. Then we mix them and then we put them in a common garden. We've done this in the past. This has allowed us to understand several principles of local adaptation, including the genetic basis. But in order to generate experiments that are really useful to making predictions under global change, we need to maximize the number of environments where we observe that evolution, because future climate changes are multifactorial and complex. So we should send it to many regions across the distribution and beyond to understand evolution in those conditions. And we should do it over time, maybe half a decade or a decade. So we actually did it, this experiment. This idea culminated in a consortium that we call GreenNet, where there is a network of crazy scientists that's endeavored to do this experiment, spearheaded by myself and 2 colleagues and 100 participants, to establish common gardens in the Northern Hemisphere. This was done by sending seeds of a high genetic diversity mixture of Arabidopsis from the 1,000 genomes, along with materials such as sensors and trays to set up 12 replicas, which look like this. These would experience the climate, something would happen, they might adapt or not, and then we would monitor them over half a decade. We have been doing that for half a decade, taking tissue samples, taking census data, taking environmental data. We have now sequenced about 70,000 tissue samples in pools, whole genome, from 32 different locations. So far, 3 years, but we have half a decade. We will release this data for your pleasure. And we've developed computational tools to be able to analyze this huge volume of new genomic evolution data. This is the analysis team, black again. The analysis team, including Tatiana Velaggio, who was giving a talk earlier. And with these computational tools, we are aiming to synthesize what happened. So what happened in GreenNet? First, it is an evolve and resequence experiment. So we have the genotypes at the beginning of the experiment. They suffer selection from the environment, and we can resequence them to see who survives and what are the evolutionary trends. Because we have whole genome data, we can monitor all the alleles along the genome, 3 million SNPs, and find parallel trends across the 12 different replicates, with the rationale that there might be drift in nature, right? But drift would cause allele frequencies to go in different trajectories. But if we find 12 parallel trajectories, that should be natural selection and adaptation. And you can see some alleles increasing, some alleles decreasing. Because we have the founder populations, we can look for unique SNPs to reconstruct the relative proportion of the 230 original genomes that we had mixed. And we can also see, in this case, how certain ecotypes or accessions are increasing in frequency in just 3 generations. This is presumably because there are adaptive SNPs, which we can look and search throughout the genome using likelihoods ratio methods that look like a GWAS. And we can identify in this one environment signals of selection, for instance, in known phenology genes, but also massive peaks in some genes that are unknown, potentially uncovering new adaptive variation. But this is not all the power of GreenNet. I'll show you some pictures. This is the beauty of GreenNet. We have evolution experiments in realistic, extreme environments, some places that it was hard to sample to. And we can see evolution taking trajectories in all of these realistic environments. So these are all the evolutionary trajectories that we've mapped in over 30 environments, which are highly repeatable with an average of 0.4. And we can see some places where drift dominates, some places where selection dominates. Okay, but was this repeatable evolution happening in the same trajectory across all environments? To address that, Xing used an approach that is called diffusion map, used in single-cell sequencing to separate the different samples based on the genetic makeup. Nobody knows what diffusion maps are, so I'm showing a PCA here, which is the equivalent linear version of it, where we can see that the founder was identical across all sites, so single dots. And over time, the different populations are expanding. So this is replaying the tape of evolution. And we can see that this evolution happened in different trajectories depending on the temperature of the common garden, with evolutionary trajectories to the right in different common gardens that were in different parts of the world, but at warm conditions. And we can corroborate this divergence through FST and typical methods. So different populations are evolving in different trajectories along a climate gradient. Are they evolving traits? To shed some light, here is the average flowering time across the 32 locations, and we see that in just a few years, a nice climb of annual temperature by flowering, with being the colder environments, later flowering, warm environments, earlier flowering, established in just 3 years. And this is local adaptation. We can see that the accessions that we know have late flowering behaviors are enriched in cold common gardens versus accessions that have early flowering behaviors are enriched in warm common gardens. So this is great adaptation. Local adaptation has happened in just 3 years. So can we predict it? Can we predict what might happen in these environments or in the future? We thought of taking a step back and see what might help us predict future evolution. From the Arabidopsis literature and plant ecology literature, we expect that typically the local genotypes are finely tuned to the local environments. And this must help us predict that if you transplant them away, they will lose fitness. And we know this from the classic papers from John Oagren, Amy Schmidt, also ourselves, and Laura, who gave today a beautiful talk about a new common garden in California. But all of these were typically contrast of local versus away. Can we, with all of these common gardens and all of these ecotypes, have something more quantitative? So we could get all the transplant distance in annual temperature from accession to garden, or pair with combinations, and then look at the fitness proxy, which is whether the accession increased in any common garden. To orient you, these are things that will be transplanted from a cold to a warm environment, warm to a cold environment. And this is what we see. The strongest local adaptation pattern that I've seen in my career. So we have here accessions that have been transplanted no matter where, things that were closely transplanted, increase in frequency, things that are transplanted far away do not. And because we have all of these accessions to environments, we can also address what are the loci that led to adaptation, to that local readaptation fast, which is work that Tatiana Bellaccio is creating using latent factor mixed models, where we associate the allele frequency trajectory with the temperature at each environment. So for instance, what we can see here is an allele that increases in cold environments but decreases in warm environments. So now we have a tool to systematically map all the alleles that might be adaptive under all of these conditions. So we thought, can we use all of this knowledge to predicting evolution, and could this be then used for genomic forecasting or genomic offsets? We thought that since this stabilizing selection theory was so strong, we could just use the environmental distance and the slope of this fitness proxy as a predictive tool, a predictive regression. So we can, for instance, hide some of the common gardens that we've run from our model, train it in all other 30 locations, and ask, can we predict the rankings of the ecotypes that are increasing or not in frequency? And the Spearman correlation is actually surprisingly high. This is the observed versus predicted trends. So moving forward, we want to utilize this stabilizing selection model to make it predictions across the geographic range. We've done this in the past using only 2 training locations, Spain and Germany. Now we have 32. And what we want to do is using all of this knowledge of rapid evolution to predicting short-term maladaptation trends. And hopefully by doing it in the present, we can actually test them. So with that, the take-homes, we've seen that evolution in this first long-term evolution experiment can be fast from standing variation. It's taking different directions depending on the climates. We can map the genes involved in climate adaptation, and we can build predictable models based on these rapid evolution experiments. And I want to thank the community that has led this. Without them, it wouldn't be possible. All of our lab and our funders and you for your attention. Thank you.
Our next speaker is Fred, and he'll be talking about network topology.
Thank you. Not so much more about gene expression evolution. There will, it's fine. There will be just a little bit of network stuff at the end. Okay, great. Thanks for taking my talk in this great symposium. And what I'm going to talk about today, the work, where should I? The work I'm going to present is contributed by people who were in my lab, but not in my lab anymore, but it's mostly contributed by Eva Koch, who did a PhD with me, and Charles, who did a lot of bioinformatics more recently, and Champak also helped with some of the analysis. Okay, so what do? We use experimental evolution in Trabolium castaneum to study the genetic basis of adaptation. Experimental evolution is a great tool to do this. I see this like a individual-based simulation in empirical context. So it's a bit of a change for me. And what is cool is that we can propagate lines, replicated lines, over multiple generations, and we can thus have time series of allele frequency changes, changes in fitness, and other phenotypes. So what I'm going to present today is an experiment where we adapted these guys to a new quote environment. Basically, it's a hot and dry environment relative to an ancestral environment to which they were previously adapted. Our source population here is the Crow line. Crow 1 line was sampled in the wild a decade ago, and importantly, it's an outbred population. As we have seen a lot today, is one of the key questions we have is how can we link these allele frequency changes or variation in allele frequency with changes in fitness, and how can we predict changes in fitness from allele frequency data? One of the challenges we have from empirical data, but also in this experimental evolution context, is that fitness is highly polygenic. We have low power often to detect what is the genetic basis of fitness changes, and this has been shown in other systems, for instance, in Drosophila also, where there is a lot of high polygenicity in the response for instance, thermal adaptation. Allele frequency also changed stochastically, especially in our experimental context where we often have rather small populations. But some of these can be addressed by having high replication, which we can do in experimental context, but also to address the problem of the polygenicity. Here, the high redundancy in the genome is to study something that is intermediate between the genotype and the fitness, and try to build a genotype to phenotype and to fitness map by studying something like a low-level phenotype, which in the present context will be a gene expression trait. So these low-level phenotype, the hope or the promise of this is that they integrate some of the redundancy at the genetic level. There is still redundancy at this level, but by having integration of the genetic redundancy into these expression traits, we can have more power to detect than the genetic basis of changes at our high-level phenotype, which in our case would be fitness. So we use, in our experimental evolution case, we use a quantitative genetic approach with genomic or transcriptomic data, and we investigate the expression to fitness relationship to evaluate the evolvability of gene expression, the response to selection of gene expression, and try to build a genotype-phenotype fitness map. To investigate this relationship between expression and fitness, what we basically do here is to estimate the genetic covariance of the trait with relative fitness, which is, by definition, the change in the mean trait value here, which is our expression trait, and this corresponds to the Robertson-Price equation, which is the prediction of the response to selection of one generation of our trait mean. I will call this the genetic selection or genetic selection coefficient, these SG values that we can estimate for every transcript in the genome, in the transcriptome. And it's directly a prediction of the evolutionary change of gene expression. It hasn't been done very often for a gene expression trait. Usually what is done is to estimate selection differential, so the phenotypic correlation between fitness and the phenotype. But here, environmental effects play here into this estimate, and so having the genetic selection coefficient is better because we eliminate this confounding environmental effect. One thing though is that this correlation, the genetic correlation between fitness and the trait, integrates all sources of selection. So it's the total selection acting on a phenotype, so it integrates direct and indirect selection effects. Indirect selection effects come from the fact that traits may be genetically correlated, and so a trait responds to the selection acting on another trait that is correlated to that trait. But cool thing is that if we estimate this genetic covariation, we can actually also estimate the G matrix of the gene expression trait, and then we can estimate the direct genetic selection component from estimate of this genetic covariance and the estimates of the G matrix. So we can then calculate our beta G, the direct genetic selection coefficient acting on a G. This is not an exponent, this 2 here, it's the footnote that you don't see down there. So what did we do? We, in the first generation for among ours, we had 7 outbred lines. We did a half-SIB, full-SIB cross with 107 half-SIB families. Each half-SIB is a male mated with 3 females, so we had 3 times more full-SIB families, and each of these full-SIB families here was split between 4 different conditions: a control, hot and dry, and the hot-dry environment. So for today, I'm only concentrating on the control and the hot-dry environments. We did the fitness assay, so we can estimate additive genetic variance for fitness, and we can do here, after having sequenced a bunch of individuals from 21 half-SIB families, we can also estimate additive genetic variance for gene expression and estimate this correlation. And so, yes, so we have RNA data from the same individuals for which we have the fitness estimates. That is basically individuals from the F1 here, and we sequence only females. All right, so we have here a distribution of the heritability in the 2 environments, CT in blue and HD in red. What you can see that for the majority of transcript, we have a high proportion of the variants in a gene expression has a genetic basis. There's a slightly lower heritability in HD. That's significant, but it's really not a lot. For our estimates of selection, genetic selection, what you can see that the relationship between the phenotypic variation and fitness variation is not as good in the HD environment than in the control environment. This could be understood as this is a new environment. The genome, the transcriptome is reacting in weird ways, and this is breaking down the relationship between gene expression variation and fitness variation. Overall, we find 55 genes in HD with significant SG after correcting for multiple comparisons. So we don't have a lot of power here to detect these significant effects. We have 75 genes if we standardize by the variance and look at the genetic correlation. What we were interested in is try to understand the effect of indirect selection versus direct selection acting on gene expression evolution. To do this, we calculated this direct response of selection from our estimate of beta G and compare this to the total response to selection. Here, these are the predicted responses. And you can see here that's a log 10 scale. You can see that the estimate of the direct selection are much smaller than the one of the total selection. So the majority here of selection, of the response to selection, is caused by indirect selection effects. And so the genetic correlation among the genes is what is really determining the expected evolutionary response of gene expression. And so since these genetic correlation, the correlation between gene expression traits is so important, apparently, then we decided to look at the gene co-expression networks. How do they look in the two different environments? What you can see here, so these are the clusters, the size of the clusters of co-expressed genes. The gray one are the genes that are unclustered. But the key thing is that we have more modules in hot dry compared to control environments. And here what is shown on this graph is the significance of the correlation of the modules with fitness variation. In the control, we have good correspondence, so we have good alignment of the genetic correlation, or the correlation among phenotypic traits with fitness variation. However, when we are in the hot dry environment, this is broken down into many, many modules that lose this association with fitness. So we think that this is more like the stress, immediate stress response that is breaking down these associations here in the first generation. And so this is based on phenotypic variation. Plastic responses here play a role in this disalignment between modules and fitness. But what we found, which is, I think, pretty interesting, is that if we take the hub genes in each module, so these are the most connected genes within each module, we find that these hub genes are more associated with selection, so they are the strongest selection, genetic selection, compared to the non-hub genes in the rest of the transcriptome. So that's good. This is giving us prediction of how these gene expression traits should respond to selection. And so after 20 generations, we decided to check whether this was the case, whether we actually can make good predictions. So to do this, we did a reciprocal transplant experiment, taking our individuals from the control and the selection lines, so hot dry, did the full transplant experiment so that we can evaluate the evolutionary response by comparing gene expression between hot dry lines and control lines in hot dry. And so we eliminate here the effect of plasticity. We did study plasticity, but this is in another publication. First thing is that we can check whether there has been adaptation or not. So this is offspring number. This is our measure of fitness. Hot dry lines in hot dry have higher average fitness than control lines in hot dry after 20 generations. So there is adaptation here happening. And then looking at differentially expressed genes, here we find 55 different genes that have significant differential expression. We don't have a lot of power. This is after correction for multiple comparisons. But what is interesting is that if we look at the response, so that the direction of the response of gene expression relative to the direction of selection that was predicted at the first generation, we find a good correspondence. So the genes that were in the positive selection at the first generation got a positive response of gene expression. They were overexpressed, and the reverse for the ones that were under negative selection. This works also at the scale of the whole transcriptome. This is also significant. We can also predict the magnitude of the response, looking here at our selected, so the ones with significant evolutionary changes. It works also rather well at the genome level. Very, very, very shortly, we looked at the change in allele frequencies. We do find a little bit of evidence of parallel allele frequency changes among our lines. This is linked with selection, our measures of selection, so the ones that are the genes with more parallelism in allele frequencies are under stronger selection. So there is a link between allele frequency changes and selection. And the hub genes are also having higher total selection, but also higher response to selection, and they are also enriched in EQTLs. And the topology of the networks do change over the course of adaptation. And I would just let you read this and maybe take a question. Thank you.
Our next speaker is Alexandre on strong selection and low predictability.
Hi, everyone. Thanks for coming. Let's jump right into it. So we already had like 2 full days about why we care about repeatability and predictability, so I don't want to belabor the point. But basically, at the core of predictability, we want to understand whether evolution is a deterministic or stochastic process. If it's deterministic, that means we can explain evolution through our known understanding of its mechanisms and cause and effect relationships, which is good for us. And if it's completely stochastic, then there's kind of no point in studying evolution, right? We wouldn't be here. And repeatability is important because it provides evidence that evolution is deterministic, at least to some degree. So is evolution repeatable? Well, over the last 48 hours, I've seen a lot of talks saying that it is repeatable. Kind of. There's also a lot of, like, a huge asterisk, right, where it's like, it is repeatable, but we know that it's not completely repeatable, right? So we know that repeatability lies on a spectrum between stochastic and deterministic forces. And how repeatable evolution is in the end is like, it depends on the interplay between these forces. And we also know that repeatability can differ amongst different scales of biological organization. So it also depends on what context you're asking about repeatability. So I think some of the main questions that we're interested in now, or at least I am, is which factors influence repeatability and to what degree? So enter the seed beetle, aka the seedle, as I like to call it, Callicebruchus maculatus. It's this cute little pest species that lives on and in these beans for pretty much its entire life. And a fun little thing about this beetle and all ectotherms is that they have an asymmetrical thermal performance curve, which means that based on constraints on protein stability, as you have very nice gentle slope leading up to a thermal optimum, and then after that you have a rapid decrease. And we can abuse that or utilize that in the lab to test questions about fundamental theories of repeatability. So because if we were to move an equidistant measure from that thermal optimum in either direction, we'd expect that the performance at hot temperatures to be much worse at high temperatures than in cold temperatures. And so selection will be much stronger at high temperatures. And because we're interested in this interplay between deterministic and stochastic forces, we expect that if natural selection is stronger, that repeatability will be higher as well. And then in these beetles, we can collect them from various source populations around the world to also test for the effects of historical contingencies on repeatability. So we have populations from Yemen, California, and Brazil. So in this experiment, we basically took these populations from around the world. They had been already lab adapted for about 200 generations each. And then afterwards, we split them up into 2 replicates into either a cold regime or a hot regime. And so in doing so, we're able to look at repeatability within and between geographic origins. And so we measured several phenotypic traits over the course of 60 generations, such as adult weight, development time, lifetime reproductive success. And you can't really see it, I guess, very well in this plot, but it basically shows the changes in those traits over the course of evolution. And then we also tested for genomic evolution by sequencing with pool-seq, and I'll explain that later. Yeah, so how do we measure repeatability? I am a big fan of this it's not super new, but this new idea of testing repeatability in a geometric framework popularized by Dan Bolnick and Stephen Delisle and a few others, where you look at evolution kind of in phenotypic or genotypic space, and if you were to take the trajectories of evolution, you could measure repeatability as the angle between these two vectors of evolution. And of course, I'm showing it here in two-dimensional space, but depending on how many traits or loci you're looking at, it could be like, a million dimensional or whatever. So this is testing the repeatability of evolutionary trajectories, but we can also test the repeatability as divergence or convergence of these populations through time by looking at the difference between the distance in ancestral populations versus the distance in evolved populations. So just as a quick little summary of what these kind of results might mean when you see them later is for angles, they are more repeatable when they're below 90 degrees and less repeatable when they're greater than 90 degrees. And for this difference in endpoint versus starting point, if it's less than 0, we're saying that these populations are converging towards each other. And if it's greater than 0, then we say that they're diverging from each other. And what's nice about these metrics is that they're kind of non-mutually exclusive. So you have a greater ability to describe how evolution is proceeding in these populations. So here we're looking at the pairwise angles between all the populations that we tested, both for cold-adapted populations up top in blue and hot-adapted populations on the bottom. And then I've separated the within-origin comparisons from the between-origin comparisons as these colored points. And what we see initially is that the mean angle between population comparisons is lower at hot populations, which means that it's more repeatable for evolutionary trajectories. And this was like the very first result that I got when I was doing the results, and I was like, yes, science makes sense, and it's easy, yes! But maybe from the talk title, you realize that this will not be the case very shortly. So then I looked at the effect of contingency on these populations, and it's clear that there is an effect on contingency. We see that usually within origin estimates are much more repeatable than between origin estimates. And also, there's a lot of variation in how different origins are on average repeatable with other comparisons. So we saw that heat adaptation is more parallel, but we also see that it's more divergent, which isn't necessarily what I was expecting as my second result. But what that could mean, just as like a visual representation, is that these cold populations, they are actually, their rate of phenotypic change is not as great as those in hot populations, and there's actually more variability in the rate of phenotypic change in hot populations as well. So you could have a scenario like this, where hot populations are kind of more parallel with each other, but they're still diverging in phenotypic space. So then I was interested in the repeatability of genomics and genomic evolution. And so here I have just an example population, and I've highlighted where outlier SNPs are shared with other populations. So Brazil in green, orange for Yemen, and then the second California replicate in yellow. So there is quite a bit of repeatability here, but I wanted to quantify that in the same framework as I did the phenotypic evolution. So what I did is I took basically all the outlier SNPs for all the populations, and I pooled them into like a set of thermally adapted related genes, SNPs, and then I did basically the same analysis as the phenotypic evolution. And then here, we actually see that there's greater parallelism for cold adaptation in their evolutionary trajectories. And even though these seem kind of orthogonal and not very different from each other, when you consider the dimensionality of the data, looking at almost 500,000 SNPs, that it's still quite a significant difference between the two of them. We also see that cold populations are converging towards each other in genomic space as well. And so if we go at the gene level, so I identified each of the outlier sets for each of these populations, and I was looking at all right, which genes are being hit by selection? What's the overlap between them? And again, we see that cold populations have greater overlap than hot populations in the genes targeted by selection. So I want to shift gears a little bit, and I want to talk about how we kind of dissected the basis of thermal adaptation a little bit. I wanted to identify different categories of SNPs which were synergistic between cold and hot populations, meaning that their allele frequency change was in the same direction. Antagonistic SNPs, where the direction of allele frequency change was different between the two thermal regimes. And then privately selected SNPs, where there was selection in one thermal regime, but not for the other. And then what we can see from that is that thermal adaptation is primarily driven by temperature-specific mutations. So if we look on the left-hand side of this, we see that most SNPs are occurring in these, or most SNPs are categorized by these privately selected thermal. Sorry, I'm really nervous. I'm going to take a little reset. Most SNPs are behaving as though they're privately categorized for either thermal regime. And then if we actually take a look at the overlap in those, we again see that the proportion of SNPs which are selected for between all the different geographic origins is again greater in cold regimes over hot regimes. And then I know that we all have our opinions about Go terms, but I did this anyway just to see. And so I was looking at Go term enrichment between these geographic origins as well, and we see that the cold had a higher proportion shared between at least two origins as well. So here I'm like, okay, this goes against what I was thinking my hypothesis would be, that hot adaptation would lead to more repeatable evolution, but we see that that's not the case across several different biological scales of organization. And actually we have some new transcriptomic results which kind of suggest the same thing on that level as well. So what are some possible explanations of that? Could it be that heat actually isn't imposing that much selection as we thought? Well, so what I did to test that is I estimated selection coefficients across all these putatively selected sites for cold and hot populations, but we see that hot populations on average have a higher absolute selection coefficient than cold populations. So I don't think that's the case. Then I thought, well, maybe it's a difference in kind of the genetic basis of these two, either hot or cold adaptation. And so I looked at the number of SNPs or genes which were targeted by selection in each of these regimes, but it looks like hot populations have a less polygenic underbasis, which we would expect to actually increase repeatability because there'll be less redundancy in adaptation to high heat. So I don't think it's that either. Could it be drift? Well, maybe. So I estimated the variance effective population size in these populations based on changes in allele frequency over time. And we see that hot populations do have a lower effective population size than cold populations, which is actually concordant with census population sizes. At high temperatures, we do see a slight decrease in population size in our experimental study. So that could be one possible explanation as to why hot populations actually have less repeatability. It could also be that there could be temperature-dependent epistasis. So we have like a little hypothetical example here based on real theoretical models of, So in here we have a reproductive rate increasing exponentially with temperature at colder ranges due to relaxation of thermal constraints on reaction rates. But we see that it starts to follow a pattern of diminishing returns at warmer temperatures due to ecological constraints, placing limits on reproductive output. So this results in strong selection on the mutation at cold temperatures for all backgrounds, as these are far from their optimal reproductive rate, but weak selection at hot temperatures across all backgrounds as they're close to their maximum achievable reproductive rate set by ecological factors. So in this scenario, epistasis is weak and selection is similar across all hypothetical backgrounds here. However, at warm temperatures, there's an increase in range of molecular failure rates, here illustrated by a loss of protein stability. And across these three hypothetical backgrounds, we have stable, intermediate, and unstable wild-type proteins, and mutations increasing stability on them. So when we introduce a stability increasing mutation amongst them, there's no effect at the colder temperatures, but there can be very strong epistatic effects at high temperatures. So, this is kind of difficult to test for in our study, but, it could be a possible explanation as to the patterns that we see. So finally, kind of what is interesting, I think, to a lot of people here is can we, instead of actually going out and measuring traits, can we just measure their genomics and say something about the repeatability there? So here we're looking at their genomic angle and divergence on the x-axis and their phenotypic angle and divergence on the y-axis. And we see that we have really like idiosyncratic correlations between them. So it's kind of not really, I don't know, we even get some weird patterns where like for phenotypic adaptation, you see this very strong negative correlation between genomics and phenotypic results. If you have a good explanation for that, let me know. I'd love to hear it. And then, so when I came to this symposium and I was reading through the abstracts, I saw the word genomic offset a lot, and I didn't know what that was like a week ago. But I read up on it, and I made a little plot to share with you guys because I thought you would like it. So I calculated the genomic offset for each of these populations, as well as a phenotypic offset, so just the distance between the reference population and the other comparison populations. And I wanted to see, it's like, can we estimate these from each other? And the answer also is kind of like, no, not really. But when we can, it's a little bit more correlated in cold populations. So yeah. So conclusions. So adaptation to heat may be more difficult to predict than we think, despite stronger selection. And of course, this has large implications for conservation because temperature is a huge axis of environmental change. It could be that demographic and possible temperature-specific mechanisms should be taken into account in predictive models. Contingency, of course, plays a role in repeatability, and we need to study further why we see these large variances between different populations, what makes them more or less repeatable. And then, of course, phenotypic estimates are difficult to predict from genomic data, at least in this study, although I've seen some promising results in other talks. Yeah, thanks.
Our next speaker, is it Giacomo?
Yeah.
Yeah, Giacomo is going to talk on environmental challenges affecting the rate of adaptation.
Yeah, thank you. Thank you, everyone. Thanks, everyone, to be here. Really excited to present you this work. Is another consortium work and a joint effort from many, many, many people. And first time I present this, so I'm glad to have any feedback on that. So, of course, don't have to convince you much today that predicting and understanding how organisms will respond to climate change is something important. And whatever is the stress that we think about, so here I'm just picking a known one, temperature, and whatever is the organism that we like or the thing that we study, whatever organism we're going to think about, they have to respond to that stress and evolve potentially to deal with that. And here again, the plot twist is already here. The story is a bit more complicated than that, of course. So organisms will rarely have to deal with just one stressor, right? So big plot twist. At least two stressors potential the case, right? It's not just temperature, it's temperature, salinity, it's humidity, is a lot of things going on. So jokes aside, so if we embrace the complexity of the environment, this is the minimum case where organisms will have to evolve and to deal with, basically. Okay? So what's going on here? Because, of course, dealing with one stressor against two, or actually two at the same time, is a different thing. And this will change our basic prediction likely. So we can distinguish, like, very simply put, like two main predictions going actually in opposite directions. So we can expect that under multiple stressor conditions, we will have a slower adaptation because of something called the cost of complexity. So again, simply put, is much simpler to deal with one problem at a time, right, than with two. Or we can actually predict a faster adaptation under multiple stressor conditions. And this is because we can expect under multiple higher selective pressure and a wider genomic target. So there's some theory, of course, behind it. It's rooted in theory, but evidence are not that much. This is 3 cases I have where actually they evolve organisms under single multiple stress condition. There are quite a few things in one generational experiment, but multiple, so real long-term evolution, there's not really that much. And it's interesting because it's kind of, I'm here inventing the wheel. I mean, of course, there's multiple stressors acting at the same time, but we don't know much. And so if you go back, if you were there yesterday symposium of predictability or evolution as well, well, this is potentially a very kind of simple question, but we don't know. So a way to push our knowledge, actually, because we don't know much. So many people in southern France just joined forces and tried to answer the simple question and trying to draw some generality for how organisms will respond under single multiple stress, or how do we evolve. And so this is the organisms that we're dealing with. So people just starting evolving these organisms are a single multiple stress. And we have, in this data set, we have phages, bacteria, some soil bacteria as well, human cells, microalgae, yeast, many ciliates, and actually 2 plants. So as I said before, it is an ongoing project, so I don't have still all these organisms. It's just I'm waiting for some data to come. Okay, and of course everyone choose and pick the stressor that considered the most relevant for their study organism, of course. So here are example of the stressor used for the experiment. We have antibiotics, copper, salt, water, or temperature. And the minimal design that we use and we discussed was, well, we'll evolve some organisms, our model system under no stressor, under single stressor, stressor 2, and the combination of the 2. Okay, and how the experiment works is, of course, you know, we have details for each of the systems. It's a bit more complicated to put, but the idea was to have a transfer experiment similar to maybe what you're familiar with, so Lenski experiment, so long-term evolution experiment. So organisms were transferred, and fitness was estimated at the beginning, and what the experimental decided was the end of the experiment. And here, when I say fitness, you're going to see an R0, R0, so it was measured as exponential rate of increase, which, of course, when we have to integrate plant, we're going to have to rethink about many other things, but that's another story. Okay, this is how it works. Just to give an example, this is Pastoria aeruginosa, one of the data set that we have already. And so fitness was estimated. What you have here is the color code that also I'm going to use throughout the talk. You have in gray or black the control condition. In blue, you have the single. In this case, Pastoria was tested and evolved under copper or temperature stress, and the combination of the 2 in red. So this is the fitness estimate at the beginning of the experiment, and this is what we have at the end. Okay, this is not going to go through these details and the result, what does it mean, but this is just give you the framework of the study. And this is what we have so far. So you can see the little cartoon as before is the organism. If it's repeated, it's because people were also able to have different strains. And yeah, this is the main result. So this is what we obtained. Not going to detail again, too complicated. But of course, now it's really time to do some stats on that. So we use a meta-analytical approach because this is basically going through literature and taking effect size and the associated error. And so I use a Bayesian framework to analyze these differences, and which allowed actually to propagate and cascade the fitness associated error and uncertainty throughout the model. And of course, I include phylogeny, stressor identity, and many, many other factors and hierarchical model, basically. Okay, let's have a look at some of the results. Okay, this is a posterior of the model prediction, the Bayesian framework. On the Y, you have density, which is the density of the prediction, so it's not real density of anything. You can ignore that. What we're interested is how, we show a lot of distribution now, how this distribution moves, basically, okay? And when distribution tend to overlap in this framework, it means that there's little to no effect. When they're kind of good, well separated, meaning that we can say, well, there's a difference, okay? So this is what the model tells us for the control. This is the fitness estimate of the control. And when we look at single and multi, of course, we tuned the experiment, and that is a stress compared to what was the standard laboratory condition. We see a reduction in fitness. So lower fitness under stressful condition, single, multiple. As I was saying, you can see some overlap there between red and blue, meaning that tend not to be different. But this is because actually when we discussed and planned this evolution experiment, we decided to use fitness as a currency, meaning that we try to tune the multiple stressor not to simply add the single stressor that we use in the single stress regime, but it was tuned to create a sort of the same initial fitness reduction, okay? In a way that they kind of started from the same fitness point, let's say. Okay, so this is the essential condition estimated by the model. And now I'm going to add on top of that the evolved condition with an arrow indicating that, well, each of the treatment, even the control, tend to respond to the laboratory selection. So even the control is not that surprising. We know that organisms tend to adapt to control conditions. So instead of sitting there, you start manipulating them. It's not that bizarre, actually. Anyhow, so you have now these fixed distributions, and you can see that all of them tend to increase towards the right. So increase and higher fitness after evolution. Now, what we're interested is, of course, the difference between the final and the initial condition, because they start at a slightly different point, right? And so if we subtract the final condition minus the initial, and we standardize by the control, we have actually how multiple singular control moved and how much the increase in fitness. And so this is the difference. And what we see is that the one that tend to increase more fitness is the multiple stressor. So multiple stressor so organisms evolving multiple stressor tend to adapt faster and evolve faster compared to single and control. Interesting. And then what we did as well, because we like trade-offs in evolution biology, is this is just the same plot, but we put those evolved organisms back to their control condition to see how now they deal to what was the original starting condition, the control, see there's a cost in evolving and adapting to single or multiple stressor. So I'm going to show you directly the differences that you have on the left. So this is the starting point as well for the control. And this time, things are slightly different. So I'm going to guide through that. So this is the starting point in gray. And what we have in what I just told you already is that the single condition there is the one that moved less. So they show lower increase in fitness. And so it's kind of similar to the control condition. But when we moved it back to control, it seems that it's the one that showed the higher increase in fitness. So it seems that single stress evolved, and when we tested back to unstressful condition, they show this time a correlated response and increase in fitness. Now, slightly different from the multiple stressor, which was the one that showed the higher response in fitness. When we move it back to control condition, well, it kind of is also, you can see, overlap with zero, so meaning no differences, and it overlapped a lot with the control. So it seems that it evolved the most, higher fitness, but little to no cost. Well, there's a problem here that is some of the organisms that were tested in their own control condition didn't match the one that tested in the evolved condition. But this is another story. So to conclude, we see evolution of fitness, and it seems that this match with the idea of a faster adaptation on the multiple stress condition. And of course, there's a lot of many other questions, other stuff I didn't show you, the impact of each single stressor, how relate changes fitness versus the initial fitness. We have to integrate a lot of new data that I'm waiting for. And there's also some genomic analysis from another postdoc that will integrate these data. And with that, I'd like to thank all the people that around this experimental evolution, the funding of Southern France, a region that is going to expect to suffer a lot from these conditions, changing condition. And thank you for your attention.
Thank you.
Yeah.
We're now going to have a panel discussion on prediction and evolution. So we have a few questions to get us started. And I'm going to let Seth take over. Yeah. Your turn.
Okay. Tagged in. So the first question that we talked about as a group of organizers is, what if we cannot understand evolution, but we can predict it? And this one came from Moi, so maybe I'll let you interpret if there's anything extra you want to say from it.
I see that we're passing the ball.
Yeah.
Yeah, I think it's more like a general question for also all the audience to participate. Like, we have seen situations where maybe through a correlational approach, we see that we can predict things, but we might not understand it very well. Some of the local adaptation, genomic offset approaches are kind of like that. We might not know, we might not have like a process-based like population genetic method, but we can tell fitness should be lower if you transplant a plant or transplant an animal, right? And we might have the reverse situations where we have like a beautiful process-based case, but that doesn't have any predictability, right? It could be maybe a specific gene for which we know de facto what is the phenotype, and we might come up with a model that's on how the gene should increase in frequency and follow a selective sweep, right? But then that doesn't happen, right?
One of my take-homes from the talks, which a recurring theme that we saw, is that sometimes the phenotype is predictable, but the genetics is not, or vice versa. And so that seemed to be a common theme that I picked up on today.
Yeah, I guess from my perspective, Moi, the thing I'm thinking about is the fear of not being able to understand but predict, that the prediction will not hold. Is that the fear? So if we were to use a prediction without being able to understand that its long-term viability would be lower, is that?
Maybe.
Yeah.
Yeah. And the other way around, right? Like it could also be that, a black box machine learning voodoo model, right? Has seen many outcomes, right? And has many predictors, and maybe it might be more resilient to unanticipated shifts, right? In an environment, right? And you can predict things better rather than if you have a process-based model, right?
Yeah.
How do we move forward? Anybody wants to jump
now we're
in?
looking
Okay.
out. Yeah,
Yes. Yes.
So I think I agree that we've seen many examples where you can predict the phenotype but not the genetic basis. And I think there are good theoretical reasons to expect that. But I'm not aware that we've seen many examples of the reverse. Did we? Or like that you can predict the genetic composition, but you don't predict the phenotype. Well, this is just a remark. But for process-based and AI, I think AI is trained by what it has seen. So I think I would tend to think that it's well established that this tends to not work when you provide it with, it's just like any kind of regression or thing like that. So at least in principle, process-based model, if you understand well the phenomenon, should be better at predicting, maybe imprecisely, but predicting things that are out of the box if you understand the process and the process are the same. But I think, yeah.
I agree from first principles, right? But also something that we had written down is, if the focus of your process-based model is a pattern that is less relevant, right? Maybe you might have unintended consequences, right, from other processes that you are not modeling, right? So it's a trade-off. Fred?
Yeah, one of the questions I have now after seeing some of the talks today is that how do you go from what we can show with these expensive experiments where you can do these very, very, very large experiments, you can show that you have some predictability. But how do you go from this to using this as a routine tool in conservation, for instance? Are we confident enough that we can go and tell them, yeah, see, it works with Drosophila, it works with Thaliana. You can do it for anything now. You just go and sample stuff and do the cheap version of what we have done in the lab.
This is actually a question I've thought about, and when you look into like the engineering literature, they're validating models to like make decisions about nuclear waste or like sending people into space. And so they have a really rigorous process for taking a model and saying, this is the steps you have to go to say that the model is good enough for this practical application. And I think we could learn from that literature in the conservation world, and you know, you have to work with people and have an a priori sort of a list of, okay, if something meets these criteria, this prediction meets this criteria when we test it, then we're going to apply it in this management context.
I think there's one other comment I wanted to say is that I agree that we may be far away from use case, but one question that we sort of batted around is this a case of do no harm, that where fear of action is more than fear of inaction? And how do we make that decision? I mean, I don't know that I have an answer to this, but I think it's something that, yeah, would you rather provide evidence, an evidence-based decision that you have medium to low confidence in, or nothing at all, and which is worse? I don't know the answer.
Just following up on that, I wonder, to increase the applicability ultimately to conservation, if we need to reframe the way we think and express the outcomes in terms of a parameter space of risk and decreasing that risk and that uncertainty, and how that falls more within principles of conservation management.
Maybe, in other words, is also what do we want to predict, right? It's connected to that, right? And what would be useful for, you know, the people that are monitoring populations, monitoring species, trying to categorize risks, right? And I think that maybe we also fall into the trap of our own experiments. It's like, okay, I can predict this thing of my experiment, like my ecotype frequency, or somebody else predicts the two traits that they measured, and right? So maybe this is also a call for standardization on what the outcomes should be to be able to validate models.
I guess it was just one of the things that you said resonated with one of the things that came to mind when I looked at your bullet list, and one of them for me, which is very important, is that the idea of being, you know, not paralyzed by the fear of inaction, but also being very humble and very honest and open about what I would call the scoping that you described very well, and to also agree that if you want to engage with applied issues is to maybe first shut up and listen to what the people really want, because we tend to be fascinated with our high-tech or latest tweak and then seeing that, oh, there are some deliverables there, but maybe we need to just shut up and listen to what they can, and based on what we know on a very qualitative and sometimes very low-tech basis, say what we think is doable, sometimes which is very low-tech, and sometimes say, well, we don't know. That would be my kind of...
Yeah, another comment I had is that several times in discussion yesterday and today, there were like a discussion of prediction means deterministic and unpredictable means stochastic. And to me, as an evolutionary biologist, I think we are very good at making predictions about stochastic processes. So it depends what you want to predict, but I think evolution will always have a part of stochasticity, but we can make predictions about that. And so I don't think we should really equate that, and we need to be clear about what we can predict or not. Maybe I cannot predict which exact mutation will happen, but I can predict the distribution of mutations that will happen in this line. So, and this might be useful as well, and I think we are good at that, but that's something we should maybe clarify in our discussion about this.
Your comment awfully calls to mind the moment in time we've all come through, which is the COVID pandemic. And evolutionary biology, in many cases, was called upon in a way that it hadn't ever been called upon before, to serve the community and to serve the world, in effect. And I think we can point to some pluses and some minuses. Pluses were the theory we had on transmission versus virulence has actually worked quite well. At the phenotypic level, we could predict that the transmission was the locus at which, or the locus, not that was where changes were going to happen. Where we really struggled was on the genomics. It's not surprising. There's a hierarchy, fitness at the top, phenotype, genotype, there's just way more things. And we're still struggling with that. But we actually do quite well with small things like viruses, at least in getting near casting, if not forecasting, as a form of prediction. And I just wanted to offer that.
Nice. What do we need to improve? That is something that I like. If we look at other fields, Katie, you were mentioning, more engineering-oriented fields like, climatologists in the 80s that were trying to build the first climate change projections. What did they do? I guess that one of the things they maybe were more rigorous in the validation approaches and so on, but also maybe the databases, right? I like the idea of Giacomo that we could potentially coordinate and do evolution experiments together, or as a community with more standardized ways to see what are the general principles, right? And maybe even test models across the tree of life. Is that something that could help?
I do have like a modeling mindset when I think about prediction. And so I think also that we could learn from the data science. And they've come so many miles in these machine learning and AI models. And I'm not suggesting we use those, but they use a framework where they have these challenges where there's these data sets and people come in and they build models from those data and they try to make a prediction about like what they're going to see in a new data set, and then there's a test on the new data set, and like whoever has the best model like wins the challenge. So I do think if we like brought a lot of these data sets together, maybe we could like have a challenge like that, right? Where you test a model on all these different data sets and see if there's anything general that comes out of that. But I think we should probably wrap it up. That sounds good. Go to the poster session. Thank you all for staying for this discussion and for coming to our symposium.